Hello everyone. Due to an increasing interest in multi-objective optimization, I would like to explain you how the genetic algorithm called NSGA-II, which is a possible solution to solve multi-objective optimization problems, is working. Let's start with an example. Imagine we want to develop an electric car which has two main goals: a very short acceleration time from 0 to 100 km per hour and a maximum possible range. The car company already has built some parts of the car, and we as a developer team can change three parameters: the size of the wheels, the power of the electric motor, and the capacity of the battery. Like you can imagine, there isn't only one perfect solution to this problem. By extending the range of the car, for example, a bigger battery is needed, which adds up extra weight. Therefore, the acceleration time to get from 0 to 100 increases. But how can we find now our best solutions for our problems? NSGA-II helps to find these. This is what the possible output solutions for different cars can look like. Every point in the plot stands for one car with an individual parameterization of the mentioned design parameters: the diameter of the wheel, the power of the motor, and the capacity of the battery. So a car over here which has a range of approximately 600 kilometers and needs 2.5 seconds more or less to get from 0 to 100 kilometers per hour has a different design, like you can see, than a car which is over here, so which has a bigger range but therefore also a bigger acceleration time. Like I mentioned before, our goal is to optimize our systems for the defined target parameters, in this case the range and the acceleration time. So we want to minimize the acceleration time and to maximize the range of the car. Therefore, there are solutions which are better than others, and there are so-called Pareto optimal solutions. The difference is that for these solutions there is no other better solution. There is just a solution where for an improvement in one discipline leads to a reduction in another one. As an example, you can see it here. Like if I want to improve the range of my car, I need to increase my acceleration time, and therefore I have a trade-off going on here. For other solutions, like if I look at the field here, which are not Pareto optimal solutions, I have a car here, like an individual. By changing the parameters, I can improve in one direction. I can increase the range of my car without increasing my acceleration time. On the other hand, I can decrease my acceleration time by still keeping up the same range for my car, just by changing the input parameters. The goal of my NSGA-II is to find out of a big possible design space I have, like I can vary all these design parameters in a range, every one of them, is to find the Pareto optimal solutions for our defined target indicators. As the name genetic algorithm already implies, the principle of Darwin's survival of the fittest is used to find the fittest solution for a defined problem. The fitness value is equal to the performance of the outputs of the systems. So in our case, our system is the car, and our outputs, like you saw before, where we get the fitness from, are the range and the acceleration time we need. And the genes which cause like the survival of the fittest are equal to our input parameters. So for examples, the wheel size, the engine power, and the capacity of the battery. And here you can see an overview of the general process of the NSGA-II. First, there is a population containing a set number of individuals. Each individual differ in its gene or parameters, and for each of them the value of the target indicators are calculated or simulated. Means I know for every individual in this population PT how the performance of my target indicators is. And so out of this existing population PT, An offspring population QT is created by mixing the genes of better performing individuals and causing mutations. The new population RT is therefore a population with twice the size of the parent population PT. Every individual is then ranked based on its performance regarding the target indicators. The better the performance, the better it is ranked. As you can see, this ranking occurs in different fronts, like here F1, F2 and so on. The smaller the number of the front, the better the performance of its individuals. I'll go more in detail on the next slide. Yes, and so the new population is chosen by picking all individuals of the best fronts until the size of the original population is reached. So like you can see here, F1 is added to the new population, F2 as well, and still the size of the new population hasn't reached the size of the old population PT. And yes, the last front, if the last front like here F3 is too big, that all individuals can get in the new parent population of the next generation. And in this case, because like in one front they're all individuals like have the same performance, crowding distance sorting is used, which I explain in detail later as well, that allows us to pick some individuals and to fill up the population of the next generation until it reaches its original size. Yes, and this explained process actually takes place iteratively, so it is repeated again and again as often as demanded, or until like the increase of the target indicators reaches a limit which I want to reach. Now let's take a closer look at the single parts of this whole process I showed you, starting with non-dominated sorting. So on the left side, I got you another point samples for you, where each point, doesn't matter which color it is, represents one individual of a population, and the colors actually mean which front they belong to. And in this case we have like our target indicators F1 and F2, and our goal is just to minimize both of them. And the goal therefore for the non-dominating sorting is to find out which individual belongs in which front. Like you can see here, for example, F1, F2 and F3. And to start, I explain you what dominating means in this context. So if one individual dominates another, means that for both KPIs, the value of the dominating individual is better or equal to the value of the other individual, and that for one of those two target indicators, the value of the individual is better than the value of the other individual. If you take now a closer look at the graph, comparing this green point here and this blue point here, so two different individuals, the green point is dominating the blue point because on one side both target values F1 and F2 are smaller or equal from the green individual than from the blue one, and on the other side one, or in this example both target indicators of the green point are also better than the ones from the blue point. On the other hand, if I would have a point over here, like which is on the same level as the green point on the F2 level, still if I would compare them, if they dominate or if the left point dominates the right point, I could do the same. And as I mentioned before, also the values for F1 and F2 are smaller or equal than the one of the point on the right. And in this case, not both parameters are dominated or are better, but one of them is. So the one for the F1 value is better than the value from the point on the right. So therefore, this point would dominate the neighbor point as well. Now that you understand how it is checked if one individual dominates or is dominated by another, we can start with the real sorting process. So, for every individual in the population RT, every individual is compared with every other individual, and it is pairwise checked if the individual is dominated by the other one or if it dominates the other one. Parallel, when we compare one individual to all others, it is counted how much individuals dominate the individual, and a list is created which contains all individuals that are dominated by this specific individual. So, resuming, we have for every individual one time a counter, how much individuals dominate this individual, and we have a list which individuals are dominated by this individual. Based on this information, we can start our sorting process. Our goal is to find different fronts. Finding the first front is therefore very easy. I just take all individuals in this population in the first front which have a counter of 0, means that they are dominated by none of the other individuals. To find the individuals belonging to the front F2, we once again iterate through the individuals in front F1. All of them have a list containing all individuals they dominate. Iterating through all of these lists, and for every individual being in one of these lists, there's a subtraction from -1 for the domination count of these individuals. Therefore, all individuals which are only dominated by individuals from front 1 have a domination count from 0. As an example, you can see here if individual D and E, which belong to front 1, both have in their dominates list the individual C. So the domination count is 2 times subtracted -1, and therefore the new domination count when generation of front 1 is excluded is 0, and individual C belongs so to the next non-dominated front F2. Taking this as an example and doing this for every individual point belonging to one of these lists from the first front which are dominated, we find front number 2 by taking all individuals having a domination count of 0. To find the third front F3, this process is just repeated one more time, and this time iterating through all individuals in front number 2, subtracting again and seeing which domination counters get to 0 and adding them up to the next front. This process is repeated until all individuals are belonging to one front. The result of non-dominated sorting is a sorted population RT due to its performance on its target indicators, and every individual gets a new rank variable containing the rank in which front it is. Therefore, an individual in front number 1 has a rank 1, and the individual, like a red individual, in front number 3 has the rank of 3. After taking the best fronts to the parent population of the next generation, one of the fronts won't fit probably 100% into the next parent population, which needs to have the same size as the population before. To keep a good spread in NSGA-II and avoid local maxima or minima instead of finding the global ones, the crowding distance factor decides about which individuals are added to the new population. The individuals with the higher crowding distance are picked first. The total crowding distance for each individual is the sum of all distances for every single target indicator. In our case, we have 2 target indicators. The front is sorted regarding every single target indicator, so one time it is sorted for F1, and in the next iteration process it is sorted for F2. And For every sorting iteration, the individuals with the smallest and the biggest value for this target indicator get a very high distance value. In my example, it is set to an infinite number, and the distance of the others is calculated by taking the distance value for the neighbor objectives. So like you can see here in the picture for the point I, we take the distance for the smaller and for the next bigger value in the front and build the delta of these, and then it is divided by the delta of the maximum value and the minimum value for this target indicator. This calculation is then added up to the already calculated distance for the other objectives. The result is that for the given front, in this case F3, all individuals obtain a crowding distance value. To now choose which individuals are going to the next parent population, the ones with the highest crowding distance factor are chosen and passed to the next population. When the sorting is done and the new population of the next generation is chosen, this population needs new offspring. Offspring is created iteratively with 3 steps. They are repeated until the offspring population Qt has the same size as the parent population Pt. At first, different members of the population are compared in a tournament selection, and the stronger one is picked to reproduce with another strong selected one. This leads to a crossover in their DNA, and finally random mutation can occur. Let's take a closer look at these 3 parts that are mentioned. Every child needs 2 parents. The parents are chosen by tournament selection. Randomly, out of the parent population Pt, 2 individuals are chosen and afterwards compared. First, the persons are compared by their ranks, and the one with the better rank is able to reproduce. So if A1 here is bigger than A2, this person is allowed to create offspring. In the case that the ranks are equal, the crowding distance operator is the factor which decides which person is allowed to reproduce. So if B1 is bigger than B2 and the ranks A1 and A2 are the same, this person is allowed to create offspring. This is done 2 times, so one time for parent number 1 and another time for parent number 2. Every time there's a new random sampling for 2 persons, and on this base the parents and the DNA are known and crossover process can take place. Crossover simply takes the DNA of the chosen individuals and mixes them. Thinking back of the car example, one gene would be the size of the wheel, another one the capacity of the battery, and the last one the power of the engine. In my example here, every individual has 6 genes. Crossover takes randomly half of the genes of one individual, like you can see here, and changes them with the ones of the other one. So, like you can see here, this is the DNA of one child and this is the DNA of the other child. So the genes here are just mixed up. Before adding these children to the offspring population and starting the iterative process again, random mutations can occur. These mutations can be applied very individually. So, like you can see here in this example, there's a random gene picked and modified. If a mutation is that it is modified by a percentage, or that you take the value of the gene and resample it completely new, this is in your own choice. Going back to our example, this could be the case if a random mutation would occur in the gene of the size of the wheel. Assuming that the wheel has a diameter of 27 centimeters in our example, the mutation occurs and sets 10% extra to the wheel size because we've chosen that it goes randomly 10% up or down. The new wheel size would be more or less 30 centimeters. And with this mutation, the child would be added to the offspring population. Yes, and this whole process I showed you is actually repeated until a certain number of iterations is reached. Like I can say, I want to do this for 100 generations and then stop and get my best solutions, or until my change in my KPIs is not really changing anymore in a significant way. I hoped that this video helped you understand how NSGA-II in general is working, and maybe you start coding your own genetic algorithm. Keep me updated, and until the next time, cheers!